Hello, everyone. Thank you for joining our Analytical Method Development and Validation for Compliant Testing webinar today. I'm excited to have Leo Schilling and Josh Rhein here with me hosting the webinar. I am Amy Knock, and I will be moderating today's session. Before we begin, I just wanted to let you know a little bit more about how the webinar will run. The webinar is being recorded, and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions. During the webinar, you can submit your questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the webinar. And that concludes my very brief introduction today. Leo, I will now pass it over to you. You may begin your presentation.
Thank you, Amy. And hello, everyone. Thank you for attending today's webinar on analytical method validation for regulatory compliant testing. Today we have the webinar divided up into 5 parts for you today. We're going to go over a brief introduction about method validation, and next we'll talk about method selection and evaluation of method fitness. We'll talk about method validation procedures next, and then we'll get into some of the details for how to perform a method performance verification. And finally, we'll wrap up with how to maintain compliance with testing needs. So next slide, please. We're going to start out today just by talking a little bit about what is method validation. So it's the process of defining an analytical requirement and confirming that the method under construction is it has the capabilities and it's consistent with the application requires. So inherent in this is the need to evaluate the method's performance. The judgment of method suitability is important. In the past, method validation tended to concentrate only on evaluating the performance characteristics. Method validation is usually considered to be very closely tied to method development. Method development and validation are very closely related. So many of the method performance characteristics that we'll discuss today that are associated with method validation are usually evaluated at least approximately as part of the method development trial. However, it is important to remember that formal validation of the final version of the method as a documented procedure should be completed. Next slide, please. So just to equip you with a little bit of background knowledge for today's discussion, just want to talk about a few definitions. First, the definition for validation is, well, not so much the definition, but the process of validation establishes the performance parameters and the suitability of a new or single validated method. And the most common issue that you'll find when looking for a method that applies to a sample type, where you might not have a direct match for a standard method would be a matrix extension. So matrix extension is when you have a sample type that doesn't neatly fall into the category of matrices that were in the original method validation. So a supplemental validation is required to include that. So next we have a method modification. So to a method modification, you make some small changes to a method that's resulting from an alternate equipment or changes in procedures that requires validation. So moving on to verification, it's very similar to validation, but distinctly different. People often use interchangeably the terms validation and verification. But verification is actually a process that ensures that a standard or official method is implemented with equivalent performance as previously established in validation. So when you're considering method fitness, it's basically confirming that a method performs suitably for a specific matrix. So as I mentioned a moment ago, if you have a new formulation that contains maybe a raw product that you've had previously tested using a method successfully, that same method might not directly apply to your new formula because there could be other interferences. And so in that case, it would be important to assess the fitness of that method for your new sample type. And if it's not suitable, then that's the time where we would need a matrix extension, and that process would be validation. Part of verification is also monitoring change controls. Changes in laboratory personnel, suppliers, equipment, those all require assessments to determine to demonstrate equivalency. Next slide, please. So next we'll just be talking about, once again, we're going to be talking about this several times today, the difference between verification and validation, because it's very common to use the wrong terminology. It's important to remember that if you're implementing a previously validated method, then you'll either be transferring that method from the reference laboratory, or you'll verify under the actual conditions of use, which means that your lab, that means under the actual conditions of use means implementing that procedure in your lab on your equipment, with your personnel. So importantly, your lab will not revalidate the method, but you will be implementing a validated method. Next slide, please. So why is method validation necessary? So the importance of measurement is the #1 reason. So laboratory testing is a critical necessity that it's used in evaluating goods, food, and feed, supporting healthcare and forensic investigations, checking drinking water, and verification of material specifications. There are decisions that are made based on the results of laboratory testing, and those can sometimes directly impact health and safety related issues. So a validated method that can reliably produce results each time you use it is critical. Next slide, please. So the next part of today's presentation is going to be taking a look at evaluation of method fitness and selecting an appropriate test method. Next slide, please. So the applicability of the test method is specified in the method in the scope section. So each method is specifically developed for an intended purpose. And so when you have a sample type that may not categorically fit into the applicable area for a standard method, it is going to be time to maybe do a supplemental validation or develop a new method. In the case where it does directly align, that's a best case scenario. So you have no need for changes and the standard method can be used as is. But it is important to make sure that the method does apply directly to the matrix. Next slide, please. So evaluation of the method and equipment and instrument range is also very important. So method selection is an integral part of the laboratory quality system. It's the use of standard methods. So standard methods are used whenever possible, unless otherwise specified by a compliance program or a customer or other specific need. Methods of analysis that are specified by law or regulations must be followed in accordance with those requirements or regulations. Non-standard methods are used in cases where a standard method does not exist and the customer has agreed to its use. There is a clear expression of quality objectives and testing parameters or criteria that are made when a non-standard method is used. Equipment that is used for the analysis should be calibrated and maintained as specified by the applicable regulations or guidance. For the method evaluation and record, any method selected for use must be appropriate for the requirements of the regulatory function, and it must be within the capabilities of the laboratory. So analytical methods are to be evaluated based on attributes such as their accuracy, precision, specificity, and selectivity, ruggedness, and other measures, and including others like practicality. There should be a record of the evaluation and the approval for its use in the laboratory by the designated officials in your lab, whether that's quality assurance or technical leaders. The method of evaluation, explanations, and justifications should be described in the record in detail. Next slide, please. The next consideration is choosing a method that is sufficient sensitivity, selectivity, and is not vulnerable to interferences. So sensitivity is kind of broadly defined as just in terms of analytical chemistry, change in instrument response with a change in concentration. And Selectivity is the extent to which the method can be used to determine particular analytes in mixtures or matrices without interferences from other components having similar physical properties, which is very similar to interferences, which are spectral, physical, or chemical interferences that can be evaluated by analyzing samples containing various suspected interferences. So you would want to make sure that you're checking every different variation of a particular formulation if you do have multiple formulations for a single product. Carryover should also be considered when evaluating blanks and standards and reference material. Next slide, please. So next, we're going to be talking to you about the procedure for carrying out a method validation, and Josh is going to tell you about that.
Thanks, Leo. Okay, yeah, so this is really, this section will be kind of the meat of all the details required for method validations. We're going to cover why and when to validate versus verify, where method development is incorporated and important. We'll talk a little bit about what parts of the method validation process is regulated, what's not, regulatory examples and fit for purpose. We'll cover some of the materials and performance criteria required for validations, statistics, bias, method uncertainty, and finally the levels of validations, where they apply, and how they're different. Okay, so method validation, there's really several reasons to validate a method. We talked about this a little bit already, but mostly because you're going to have non-standard methods. You have research and development methods. We have lots of modifications nowadays to standard methods as technologies and sample prep approaches have changed, extensions to existing validations, or of course to demonstrate that a new method is fit for purpose. Only methods that have been developed to the extent that it meets the needs for the purpose of the testing should be validated. And candidate methods should be investigated using experiments to demonstrate proof of concept. In other words, it really doesn't make sense to start a validation until you understand how that method is going to perform on your product. And that's where that method development and feasibility plays an important role, or sometimes I call that pre-validation, plays an important role in the process. Okay, so method development, really it's required for any sample type that cannot be analyzed using an existed validated method. Not unusual for method development to involve work on multiple approaches simultaneously before eventually choosing the best fit. Regardless of how much effort has been invested during method development, there's no guarantee the method will perform adequately during validation. So there's always risk involved when you go through the method validation in terms of resources and potentially having to redo it. But the more work that can be done on the development or feasibility or pre-validation, whatever term you want to call it, it's going to provide confidence that the validation will be successful. Okay, so is method development a regulated activity? Proof of, I would say, so there's a line that's drawn for that, right? So the first two pieces, proof of concept and method development, that's not regulated. That's going to be outside of the regulated and protocol environment. Typically, during proof of concept and feasibility, you want to have assigned personnel working on that understands what methods are best for what matrices and whether we're doing an impurity method or an assay method. You're also going to look at verifying system suitability during this phase and what is practical for the approach. And then when you hop into the method validation process, that is going to be regulated. Document validation plans, including acceptance criteria. You need to define those acceptance limits and system suitability. Perform the evaluation experiments, including robustness, and document all of this in compliance with the applicable regulations. All of this will be under QA oversight, and really can't be hidden from the regulatory process. Routine analysis as well, method transfers, define the document and process review cycle interviews, monitor performance, maintain adequate change control, and perform regular process inspections. Okay, so next we'll talk a little bit about kind of the regulatory requirements. It can be a little bit complicated. The GMP regulations were developed to apply to many different industries. So interpretation of the regulations can take some careful considerations on how to apply directly to the work being performed at your facility. You know, really the devil is in the details on this. It's critical to refer to the appropriate guidance that is applicable to the requirements for the results from testing. In the United States, Congress passed the Pure Food and Drug Act in 1906. It became illegal to sell contaminated food and drug products, which led to the formation of the Food and Drug Administration. Congress passed the Federal Food, Drug, and Cosmetic Act in 1938, which required companies to prove that their products are safe before selling them, and gave the FDA the authority to regulate manufacturing the sale of food, drugs, and cosmetics. GLP regulations that govern the development approval of the use of regulated products were not introduced until the 70s. Really, you know, the details not included in this timeline for this legislation include events that required a response for change. And the requirements for regulations developed separately by the FDA, EPA, USDA address specific and separate issues in industry where intervention was necessary to protect public health and safety. So, I mean, really, I mean, when validating a method, it's required to follow the guidance and regulations that are directly applicable to the specific category of the product, because they're developed separately for cause and may have but critical differences for certain requirements. So, really, in other words, the way you handle a submission for an EPA validation for approval, it can be very different than the USDA and the FDA, and whether this is a raw material or an API or a finished injectable product or something like that. Really, it really depends and needs to be fit for purpose. Okay, so validation requirements. Although the specific regulations have requirements that impact method validation activities, there's a foundation in good scientific practice that is generally conserved across all agencies. The slide illustrates the workflow that is essential for any method validation, right? You need to start with kind of a plan, understanding what the objective is, work through some of the method development and suitability and feasibility steps. And then, of course, there's a lot of documentation and training, verifying that your equipment is suitable and calibrated and qualified. Okay, so lots of different materials that can be used for method validations. I'm not going to go into too much detail on this, and every depending on what's available and what the product and test method that you're looking to validate and the availability of some of these materials may change what is utilized in the actual validation. Blanks are really important, right? So reagent blanks, you want to make sure whatever it is you're measuring isn't a contaminant in the reagents you're using. Probably more importantly after that is when you're evaluating active substances in let's say a finished product, you want to be sure that all of those other excipients and components in the product don't interfere with the active analyte. So running what we call a kind of a matrix blank that has everything there, but the active that you're looking to measure is an important step. So for reference samples, it's always going to be ideal when doing a validation to have certified reference materials, but that's not always the case. Sometimes we have to make our own as a laboratory control sample. Sometimes that's as a blind or handled internally. Fortified samples. This is really a critical piece to the accuracy and precision parts of the study. You need to take and have spiked blanks. Well, sometimes it's not possible to get blanks, a complete matrix blank. So at that point, you need to do standard additions and see how the response is when compared to the incumbent product. Incurred samples contain a concentration of the target analytes that is inherent to the environmental conditions. This may be soil that contains heavy metals from wastewater that is illegally managed. It could be edible tissue that have residue levels of accumulation for a veterinary drug. Rosemary contains carnosic acid, a potential antioxidant, right? That's important too. Incurred samples have to be acquired by or provided to the laboratory for method validation and process controls, and they can be fortified with a known quantity of the target analyte to investigate or monitor analyte recovery. Reference materials, certified reference standards. I'm not going to get into too much detail on that. So next we have method performance characteristics. These are the typical performance characteristics that are for chemical methods. Some qualitative methods and identification methods will not share all of these parameters, but generally this is what's going to be required and listed out in any regulation you look at, whether it's the USP, it's ICH, EPA. They're all going to talk about selectivity, and selectivity. What we really mean by that. Oops, pop back here. Selectivity is my method selective for what I'm looking for? If for example, we were talking about this before, if my matrix blank that contains all my excipients gives me a positive response in my HPLC chromatogram at the same retention time as my active API, that's not going to work. We have to show that it's selective. Sensitivity is always important. Depending on whether you're looking at a contaminants method where you're looking for low levels, or you're just trying to quantitate an active ingredient. That the limit of detection versus LOQ would be important. Analytical range, that just defines where you can apply the method at what concentrations. Accuracy, what type of recoveries do we expect to see? How accurate is the method? Precision, that really encompasses repeatability, intermediate precision, and reproducibility. How does the repeatability over multiple days, different instruments, different analysts, and that also ties into robustness. And robustness really can be expounded to mean a lot of different things. In a verification process, robustness may just be something where you have two analysts to make sure that in your laboratory both analysts can perform. It may involve when you're validating a completely new method, that may involve really pushing the limits of your mobile phases and other conditions that may come up at some point during the analysis in the future, and you want to make sure you address those items. Statistics. So statistical methods are utilized for summarizing data appropriately for making objective judgments. So validation is objective, right? We have to have a way to understand whether or not our validation passed, and that's all set by criteria that are set forth before the analysis starts. It's written into the protocol, it's approved and signed off on, and what you do then is you're evaluating things like precision as relative standard deviation and defining that coefficient of variation. Accuracy is generally done as a % recovery. LOD, LOQ, bias, these all require statistical calculations, whether it be signal to noise, standard deviation of replicates from a standard measurement. Determination of bias. Bias is the total systemic error, and there may be one or more systemic error components contributing to the bias. Bias is calculated as the difference between the expectation of the test result and the true value or accepted reference value, and can be expressed in absolute terms as a % or as a relative to recovery from a fortified sample. Method uncertainty, it's complicated. Measurements or method uncertainty is not typically calculated as part of the method validation, but it can be a useful parameter for monitoring of routine testing. However, their contributions to method uncertainty from a step in the sample lifetime that has potential to influence the result. Some of the influences affecting the result happen before it reaches the laboratory, and it may be appropriate to consider only the influences that directly impact the analytical procedure and analysis. So really, in other words, the question sometimes can arise is what is the actual method uncertainty in a controlled environment versus things that are uncontrolled from, say, sample prep or homogeneity of the actual product. It's really different. So for the validation process, Linearity and range. Calibration really determines the relationship between the observed analyte signal generated by the measuring detection system and quantity of analyte present in the sample, right? So this is typically done through calibration curves. You have known amounts, whether you're using internal standard or not, but allows you to measure against something externally. The analytical range is really just where does that calibration stretch? Accuracy, talked about a little of these each in the summary slide, but this is the closeness of agreement between a test result and the accepted value. So when we add in a particular amount of analyte, how much do we get back out? It's really kind of the trueness. Precision is the closest of agreement between the numbers. So if I measure something repeatedly, how tight is that relative standard deviation? Allows you to understand what type of variability to expect between different measurements. Selectivity, again, talked about this a little bit. Just you want to make sure whatever you're measuring is only measuring the compound that you're targeting, and nothing else is impacting that. Specificity in quantitative analysis, specificity, the ability to measure the analyte in the presence of components. So it's really very similar to selectivity. Interference, positive or negative response. We talked a little bit about positive interference already, but negative response. A lot of times, the components can suppress the response from whatever technique you're using. Important to measure that, and a lot of times you can see that in your accuracy studies. Sensitivity, it's change in instrument response that corresponds to a change in the measured quantity. So obviously, as you go down in concentration in your procedures, you're going to expect that response to decrease. And typically, as you go lower in concentration, that variability in the measurement increases. So generally, when we talk about LOD, LOQs, and LOD is going to be generally defined as 3.3 times the standard deviation, and the LOQ is 10 times. So when we, or another way to look at it from a chromatography standpoint is signal to noise. So 10 times signal to noise is generally your LOQ. Robustness and ruggedness. So robustness, measure of the capacity of an analytical procedure to remain unaffected by small but deliberate variations in the method parameters. So for example, if your HPLC, you have set to have the column conditions at 25C, you may want to push those conditions to be 20 and 35 and see how much it changes your sample. Probably a better example would be adjusting your mobile phase pH slightly one way or the other and seeing how that impacts the analysis. Ruggedness, really ruggedness and robustness are often interchangeable, but ruggedness typically relates to the repeatability as it's measured and is a measure of the reliability of or stability of the method against extraneous influencing factors. Example included change in personnel or instruments, using different instruments over different days. Okay, so who performs the method validation? After a method's developed, the laboratory should document the research method in sufficient detail that it can be included in the plan for validation. For chemical methods, the single laboratory and interlaboratory comparison studies are acceptable approaches for analytical method validation. The extent of the validation requirements will also be determined based on the intended use of the method. In some cases, including emergency situations, it may be necessary for exceptions to some validation process requirements. If there are excursions from applicable guidance or regulations or deviations from the plan or protocol, all examples should be listed at the beginning of the validation report. So levels of validation. Several references refer to levels of validation. Consistently, the requirements range from a minimum of 1 single day of experiments with one analyst and one instrument study to a full collaborative study between different laboratories and different organizations. The levels defined on this slide are used by the AOAC and FDA and it's generally accepted that the results produced from the methods that comply with the higher level of validations are more reliable, right? So level 1, single laboratory validation. A lot of times these are internal methods or customer methods that aren't going to be used outside of the scope of that single laboratory. Level 2 really is just a little bit more comprehensive beyond that, looking to kind of performance criteria that comply with an applicable validation standard, something like the AOAC appendices that define exactly how the acceptance criteria are written. Level 3 is going to be multi-laboratory, one collaborating laboratory in addition to the original. Just gives some confidence that different labs can perform adequately. And this is, that's important if the methods are going to be used by multiple laboratories. And then of course, level 4 is going to be something that's, AOAC published or ISO, USP, something that's going to be compendial and used widely. So approaches to method validation: when a method is needed but not applicable validated method, standard or official method is not available, a laboratory will plan a single laboratory validation, either because the request is specific to a purpose where there is no general interest, or because there is some value associated with the method as intellectual property. If the method is suitably validated to the extent specified in the applicable guidance, then it's possible for single laboratory validation methods to be utilized for producing results used for regulatory purposes. So, generally, you'd prefer to use a compendial method or something that's available, but this approach usually is limited to where there's no other option. A collaborative study or interlaboratory comparison are the most generally applicable for a standard method. Collaborative studies are multi-site studies that are directed by a pre-approved protocol and have defined procedures and acceptance criteria. Contributing laboratories submit a report and results are evaluated against the acceptance criteria. Results from a successful method trial are published and the new method is considered to be fit for purpose. After the collaborative study, the method may be implemented by a method transfer, which includes a comparative study between the reference and participating laboratory or laboratories, or by completing a compliant method performance verification, which we'll talk about more later. Documentation required. So the analytical procedures should be described in sufficient detail to allow a competent analyst to reproduce the necessary condition and obtain results within the proposed acceptance criteria. Minimally, these items or activities need to be documented in the method validation report. The specific documentation requirements depend on the applicable regulations or guidance for the testing, but the items listed in this slide include the critical components that should be included for most validations. So when we look through some of this, really a validation plan or protocol is going to be tied into all of those systems, software, equipment, and everything being qualified. You've got to have SOPs and training. You need to have system suitability established. Okay, so for the validation plan or report, the validation work is performed and the results reported is directed by the validation plan or protocol. The general requirements of a validation plan or protocol and validation report are often included with applicable guidance documents and can be used as a template for documentation. Common and critical parts of the validation plan include a unique title. For chemical methods, the title should identify the analyte, method, and applicable matrices, and perhaps even the technique used. Scope. You want to have a scope there that talks briefly about the information about the method and a short description about the method, as well as the details on the status of the method. Is it an international standard? Is it an in-house method? You want to have the analyte measurement unit, types of sample, intended use. The experimental design is also important. This section should also outline the purpose, which may be a single laboratory validation of a new method, verification of performance of a standard method, or an extension to the scope of a validated method. The performance characteristics which will be or were investigated should be described as well. And really important is the acceptance criteria. And that's really kind of what's listed here on the screen. This section should give us specifications for the performance characteristics that will be used. So what are the acceptance criteria for your accuracy, precision, specificity, so on and so forth. Usually it's going to be in a table format like on the screen. Summary, results should be summarized in data tables, including statement as to whether the method is fit for its intended use or should be given in the report. The last section of the report should summarize the validation work and the results. However, it's not uncommon to include an executive summary at the beginning of the report that provides the essential details and the outcome of the validation. Okay, Leo, I'll turn that back over to you.
Thank you, Josh. Next slide, please. So next we're going to talk about the method performance verifications. And as we've talked about a few times now, there's some common misusage or use as synonyms of the terms validation and method verification. So we've reviewed now all the components of a validation and talking again about the difference here. One way to understand the difference is to accept that a properly validated method does not need to be revalidated. If there's a need to extend the validation to a new matrix or an analyte, then the outcome is a supplement to the original validation. So an example there would be if you have a new formulation, you would need to revalidate if there are different components in that mixture. So, and the reason that is because you could have potentially different interferences in the new formulation versus the other ones that have been validated. So that's not a performance verification, that would be a supplemental validation. Also, I guess, and if no changes are intended, when implementing a validated or standard method in your laboratory, then, it should be sufficiently verified. So some laboratories call this activity a method performance qualification, but it should not include the evaluation of robustness under typical circumstances. That brings me to another thought where, if you have a method that is validated and you have a client or a customer or some other inquiry asking about accreditations, just because you have a validated method doesn't mean that it's accredited by an accrediting body. To have an accredited method, you should have your validated method, whether it's a custom method or a standard method, it should be validated in place in your laboratory. The procedure for getting a method accredited involves an inspection from an authorized accrediting agency. So they would come out and they would inspect to verify that you are performing the method as written in your SOP and as required by the standard that you are seeking accreditation for. Next slide, please. So I thought, another nice way of, nailing down the difference between verification and validation would be just to, show you the differences in a kind of a visual format here. So, this is showing the differences between verification and validation for both quantitative and qualitative methods. And if you look down at the, bottom part of the screen, you can see that, pretty much the only thing missing from validation is the calculation of uncertainty. If you flip over to verification, you're really not going to be too invested in checking on reproducibility, and you should not investigate robustness. For a validated method, robustness has already been investigated, and unless you're doing a supplemental validation, and it might be appropriate for you to do an investigation of robustness, but under normal circumstances, investigation of robustness is pretty much the only difference for a qualitative method between verification and validation. Next slide, please. Just wanted to give you an example here too. So we have a case study to present for you where, there was a request for use of a standard method. So, when you have a request for a certain type of testing, you should take a look at, checking the references, review the original validation compliance with ICH-Q2. Perform a gap analysis and perform missing validation steps prior to the transfer method performance verification process. You should do a base acceptance criteria on the current validation studies. Document and describe the transfer or verification process in a plan or protocol. Investigate the deviations from the protocol and document comparative outcome in the transfer or verification report. Next slide, please. So the challenge that we had for this example was that a medicated feed manufacturing facility was required to be licensed by the FDA to produce feed containing veterinary drugs. So part of the requirement for being licensed is that products that are produced must comply with the specification for accuracy of label claims specified in 21 CFR Part 558.4. In order for our laboratory to provide the testing, we needed to implement a method of analysis that's capable of producing results that will be accepted for regulatory use. So as we've talked about a few times now, the first thing you want to do is check and see if there's an existing method that is applicable to the sample type. And if there is, you want to use that first. So in this case, AOAC official method 2006.01 meets this requirement. It was selected for evaluation. Next slide, please. So FDA guidance for industry number 135 defines the requirements for the validation of analytical procedures for type C medicated feeds. Type C feeds are complete diets for animals that are intended for continuous feeding, but they contain a single or a combination of veterinary drugs at therapeutic concentration that's been approved by the FDA for a specific use. Because the FDA has approved this drug for multiple uses for multiple species, it was necessary for the laboratory to verify the method performance in several different matrices. Note that it's also important to consider, again, take a look at what the regulatory requirements are. They were all developed separately and independently, so there are very small differences. So it's important to be familiar with what the requirements are for your specific purpose. There might be differences in qualified personnel requirements or equipment, data systems, things of that nature. Next slide, please. Finally, the solution. After the completion of the method performance verification experiments, the data was summarized by the laboratory analyst, reviewed for accuracy and quality requirements by a scientist, and then assembled as a report, which is distributed for review and approval throughout the laboratory. It's maintained in compliance with local SOPs and strict change control. After a quality control plan is defined and process control checks like trend charting for system suitability parameters and quality control samples are implemented, the method is ready for regulatory compliant testing. Next slide, please. Finally, we're just going to be wrapping up the presentation today by talking about how to maintain compliance. Next slide, please. So just using validated methods, you can't just call it a day. When you have a validated method, there's maintenance and there are certain requirements that are necessary when you're using a validated method. Also note that research methods that have not been validated may be suitable for investigational purposes, but be sure to not use those research methods that have not been validated for any testing that is subject for regulatory compliance. So just want to call that out. But to implement the validated method, an SOP or test method description should be adequately describing the sample preparation analysis procedures. However, it is critical to evaluate the validation to ensure that it is fit for purpose before implementing it. That also comes back to evaluation of method fitness. So just the take-home message here is to try and anticipate if you'll receive samples that are outside of the scope of what your current plan for your validation is, and considering adding those to your plan, or just do plan for supplemental validations as those new matrices come through. Also, some guidance and regulatory requirements specify more than just a validated method for compliant testing. You may need to have specifically qualified instrument systems, and computer data systems, a validated LIMS system, qualified personnel, and process controls and monitoring. A critical example of process control is knowing the bottlenecks and limits for the testing in your laboratory to consistently use the method. It's important to know how many samples can be tested at once. Next slide, please. Next, just finishing up, we've talked about this a couple of times now, but the documentation of validated methods is important. So the method subject to validation is performed using a documented procedure, which should be considered draft until the validation report is approved. Once the validation process is complete, it's important to document the analytical procedure so that the method can be clearly and unambiguously implemented. There are many reasons for this, but importantly, it's critical to make sure that you're producing consistent results. Next slide, please. So using the validation data to design a quality control plan is a critical part of the validation data to ensure that the results that you're producing are reliable. So tools that you can use for this include flowcharts and work instructions, job aids, checklists, control charting, process inspections, phase inspections. But mainly it's important to just make sure that all of the controls are in place to make sure that you're monitoring the quality of the results that are going out of the laboratory. Next slide, please. So this finally brings us to calculating and reporting results. Some very important details here. After the validation is complete, you should have some data that's appropriate for using to calculate method uncertainty. We talked about measurement uncertainty earlier today, and there are many components that go into that. So it might be appropriate for you to consider only the influences that are directly contributing to bias on the test system. So the sample preparation procedure, the analysis procedure, and things of that nature. So everything that happened before the sample reached the lab, you may want to consider excluding from the calculation of measurement uncertainty because we're completely outside of your control. Not detected results should include the LOD or the LOQ or some other descriptive information. A lot of customers will interpret not detected as absent, which is not the case. So it's important to make sure you put the limitations of the method on that report or in that certificate of analysis. It also may be appropriate to include the reference method or the method ID on the certificate analysis or report. And also, if there was any type of modification or deviation, it should be, first of all, those should be noted. There should be traceability for compliant testing. So any modification or deviation should be noted in detail in sample preparation. It should also be identified on the certificate of analysis and report. This also includes requests to extrapolate an estimate for a result that's outside of the quantitation limit. So you'd be surprised by how frequently a request to report something outside our analytical range is requested. So if you agree to do this, make sure that it's clearly noted on any report or certificate that it's an estimated result and not a quantitative result. Next slide, please. So these are some, this is a handful of references. This certainly is not an exhaustive list. I kind of went to my guidance document folder, and picked out the ones that I most frequently reference myself. But all the there is appropriate guidance and regulation out there. Make sure you don't just pick one at random and think that they are the same. That couldn't be farther from the truth. The regulations have specific purpose for all the things that are specified and required in them. So it's important that every regulation is followed to the letter. If you have questions about that, you have, I'm sure, experts at your disposal that you can contact, including those of us at Eurofins, if you ever need any assistance with choosing an appropriate guidance document or regulation that applies to the testing that you need. Next slide, please. And if you're joining us today and you're thinking to yourself that you need a new method for an animal product, I hope that you'll be able to join us for the next webinar that we'll be giving. It's going to be navigating the product development process for animal veterinary products. It's going to be presented in December. We don't have a date yet, But if you would RSVP to info@eurofins.com, we can get you that information for the date. I'll be presenting that along with a colleague, Carrie Nichols, who is one of our analytical services manager at the Eurofins Nutritional Analysis Center. So that should be a really good and informative presentation. Next slide, please. So that brings us to some questions, and I think I saw quite a few coming through the chat. So Amy, do you want to kind of help us out with that?
Yeah, that would be great. I know we have received a number of questions that have come into the chat. As we mentioned earlier, if for some reason we don't get to all of those questions, we will be following up with a message. Also, our responses might be brief here, but if you have further questions, please don't hesitate to email us. So it looks like we have quite a few here. I will just move into them for the last remaining time that we have. The first one that has come in, what is the relationship between method robustness and ruggedness?
I can answer that one. So those terms are typically used interchangeably. I think the quickest way to answer that is that method robustness are those characteristics that are intrinsic to the analytical procedure. So if you are extracting a sample and you have a shaker that's agitating the solution, a robustness experiment would be to set it for the 15-minute time that's specified by the method, and also a shorter amount of time and a longer amount of time to see how that change affects the outcome. So that's robustness. Ruggedness would be something that I would consider to be outside of the test system. So maybe a different instrument, the test method. So maybe a different instrument system, or a new analyst, or a different piece of equipment.
Great, thank you, Leo. The next question that has come in, what are the 3 types of method transfers?
I can do that one too. So there are different types of method transfers. USP 1225 describes a type of transfer where it's a revalidation or partial validation. So that's one type. There's a co-validation of laboratories, where the transferring laboratory works with the receiving laboratory in an interlaboratory validation effort to transfer to the participating laboratory. There's also a comparative study type. And the analysis is conducted on samples from the same lot, and the laboratories that are participating will have those results compared to each other.
Great, thank you. The next question here, what are the key steps that should be considered on any pharma, ingredient, excipient, dietary supplement, analytical method transfer checklist?
Ooh, good one. I'll take that one too. The receiving laboratory should verify that all the equipment and systems that are required to perform the analysis are available and qualified.
Great, thank you. What is the most accurate way to calculate the LOD and LOQ?
That's a loaded question. The simplest way of doing it is, Josh, you want to take that one?
No, I was going to say, yeah, I think it really depends on the actual method that you're validating. So, if you're doing chromatography, I think signal to noise is really important. If you're doing a titration, you want to understand what the drop size is in your titration vessel, or what the minimum delivery volume is. And then, of course, one of the most overarching ways to do this is to evaluate the standard deviation from a known standard. Leo, if you want to add anything to that.
Yeah, I mean, I guess I'll just emphasize that there are multiple ways of doing it. The one that I think pops in my, the little equation that pops into my head is, I think, from high school or college or something like that, where it's, 3.3 times the standard deviation. I mean, I remember thinking, the standard deviation of what, So is that going to be the standard deviation of the residuals from the standards, or So there are quite a few questions there. The simplest way is like I said, 3.3 times the standard deviation for the LOD, and 10 times the standard deviation for the LRQ. But I think the most important critical thing when calculating LOD and LRQ is, not so much obsessing over which method you choose, but to pick one that's appropriate and describe in detail why that method was chosen and how it was calculated in the method validation report.
Great, thank you. The next question here, what do you need to do a validation or verification in your method in a matrix sample reformulation?
Josh, you want to take that one?
Yeah, sure. So really, if you're reformulating, it's essentially it's an extension of the validation. So, those pieces really under the performance verification are likely going to apply. In particular, is the selectivity going to be impacted by your change in formulation? Number one, and then number two, are there going to be any matrix effects or suppressions? So that accuracy study, the accuracy and precision is going to be really important along with that selectivity. Your LOQ shouldn't change. The range of analysis shouldn't change. So really, those are the key pieces for that.
Great, thanks. And it looks like we have about time for 2 more questions here. So the next one that has come in is, what is the difference between validated and accredited method?
Leo, you want to take that?
Sure. I've mentioned that briefly in the presentation, but an accredited method means that, and you could actually have a method accredited that, that's not true. So an accredited method would be a validated procedure that you have established in your laboratory, where an accrediting body will come out and do the inspection of how you complete the analysis from start to finish, and make sure that everything that you're doing is, what you're saying you're doing. So, for example, for ISO 17025, if you want to have an ISO accreditation for a test method, there would be some qualified, accrediting agency that would come out representing ISO and would evaluate you against the standards for 17025.
Right.
And go ahead, Josh.
Can I add to that, Leo? Yeah, I think it's really it really speaks to the environment in which you're trying to comply. So, right, the FDA may not necessarily come accredit your method, but you need to be ready to defend the validation in the same fashion that the ISO accrediting body would come out to look at your validation and your systems and your training and provide you with that accreditation. So I don't think there's necessarily a change in how you would approach the validation. It really pertains to the regulatory body that you're aiming to appease.
I'll add to that too. There's a different, and we get this with our European clients. Our European clients will ask us for our GLP certificate, and the FDA actually doesn't do facility inspections to approve them for GLP work the way it's done in Europe. So I mean that's one difference that you might see internationally between accreditation. So if you are coming from Europe or have experience doing work with a laboratory in Europe, don't be disappointed or think that you have substandard quality from a laboratory in the US because we don't have a GLP certificate. It's just not something that the FDA performs or allows.
Great, thanks. And I think we have time for this one last one here. How much training on the specific method should be done for a chemist participating in either intermediate precision or method transfer when they have experience in similar methods?
I can answer that, Leo.
Okay.
So really, I think in that particular case, it's just important that you follow your internal procedures and whatever your SOPs define as as being trained. You know, I know exactly, you know, what you're saying is, you know, if I have an HPLC analyst that is trained in a bunch of HPLC methods, however, we develop and are validating a new method, how could that person be trained in the new method? You know, in that particular case, the piece of the validation is going to help essentially validate that. But you want to make sure that they have the appropriate, you know, if there's a certain sample prep technique that is that's different than what they've done historically, you want to make sure to document some way of evaluating that. But those precision samples that they would run as part of that intermediate study would fulfill that.
Great, thank you. And it looks like that does wrap up our Q&A session. We are coming to the top of the hour. I know that a lot of questions have come in. As a reminder, yes, you will receive a copy of the slide deck as well as a recording of the webinar, probably within 3 business days. And again, I want to thank you, Leo. Thank you, Josh, for your presentation today. And thanks everyone for attending. Have a great rest of your day.
Thanks, Amy. Thank you, everyone. Bye.
Thank you. Bye.